So next, we're going to talk about the structure of DNA itself. As we know, Watson and Crick used the results of some other folks' research and some clues that had been generated by Rosalind Franklin, who suggested that the molecule was helical based on X-ray crystallography. And Edwin Chargaff had noted that the ratios of A to T and C to G, and those are the four bases that comprise the sort of middle of the molecule, are equal. And so there must be some kind of pairing that went on. And so, as you've seen in some of the videos we've looked at, Watson put all the pieces together, and along with Crick, sort of published the structure of the molecule. And we'll read that classic paper. And the way basically that DNA works is that there's a sequence of linear information if you read along the molecule. And so, I guess the genetic molecule needs to be able to do two things. It needs to be able to be replicated across the generations so that, genetic instruction can be passed down through the generations, and it needs to code for proteins. And proteins are the molecules that enact, our development, our physiology, that sort of make life happen, mostly through enzymatic activity, though proteins are also important messengers and structural molecules. So, how is DNA replicated? Well, we'll dig into this in a lot greater detail, but the idea here is that each half of the molecule, each strand of the molecule, if you separate the molecule down the middle, can be used as a template for a new strand. This is called semi-conservative replication, and Watson and Crick initially suggested that this might happen because they noticed how the molecule had this symmetry, how one side of the molecule had a sugar phosphate backbone with a base hanging through the middle, and how that base was complementary to another base. So if you take half the molecule, you can generate the other half. And so this is a great explanation for how it is replicated.